and welcome. We will continue our discussion on interaction of light with biological matter. Previously, we discussed interaction of light with molecules, biological molecules that is. Then we discussed a bit about interaction of light with cells. So, as a natural progression, in today's lecture, let us discuss interaction of light with tissues. Now, before I go forward, there are couple of mistakes that I need to owe up to and I need to clarify. I have divided the mistakes in three separate categories. The first one being some of the words that I am mispronouncing. I have this bad habit of jumbling up two words together, and say for example the word prevalence, I pronounce it as parvalent, which is wrong. This becomes a nightmare when I am trying to describe the names of several scientists. So, Watson and Crick model, I think I mispronounced it as Kark module, and Krebs cycle, I think I mispronounced it as Karp cycle. So, these are some of the pronunciation mistake that I apologize for. That should be corrected. So, the correct pronunciation is important, especially when we are talking about names of people, names of eminent scientists. The second mistake that I usually do is oversimplifying certain things. So, this technically is not necessarily a mistake, but you understand that this is an interdisciplinary topic. So, like for example, when I am discussing about the fundamental difference between an organic compound and a semiconductor material, I try to discuss it as organic compounds have thick, those HOMO and LUMOs, and there are several sub-bands from which electron can jump, both internal conversion, etc. Semiconductors, usually I have described them that electron simply go from valence band to conduction band, drops from conduction band to valence band. Those of you who are students of advanced semiconductor know that that is also not the case. It is also possible to have quite thick bands and sub bands inside semiconductors, inside valence band and conduction band, and non-radiative transition between different parts of conduction band can happen. These things become especially true if we are looking at quantum dot semiconductors or quantum well or quantum wire semiconductors. Similarly, when I am discussing biology, I simplify that nucleus contains DNAs and RNAs, and outside the nucleus there are proteins, but many of the life science students will correct and rectify that DNA is also present outside the nucleus in mitochondria, or there are certain proteins present inside nucleus. So, see, I purposefully do this simplification. The idea here is to not to confuse people. If I give on talking about all these exceptions, all these things that are usually outside the norm, all those things which further complicates or confuses things, I don't think it will serve any purpose, right? If you are specializing on, say, cell, or if you are specializing in quantum physics, or if you are specializing in semiconductors, it is quite okay for you to go into further detail of that. I am simply giving you a glimpse, especially to a vast interdisciplinary group of students who come from various backgrounds. So, these information usually does not increase your knowledge, it simply confuses you. So, we will continue with this simplification unless you tell me in the comment section that you are terribly upset by these some of these things that I am not mentioning, or I am overly simplifying things. The third mistake that I do is I misspoke. Sometimes I believe I say that the electron absorbs the photon and the electron goes to upper level excited states, and the photon returns back to the ground state. Photon does not return back, it is the electron return back to the ground state after emitting photon. So, see, neither are we actors, nor I have a teleprompter, nor I have memorized a script. Whatever I am saying is an extempo. So, it is possible that couple of times there will be certain words that I will misspoke. Like I think in the fluorescence part, while discussing about quenching, I discussed molecule B is colliding with molecule B, molecule A is colliding with molecule B. So, you have to be sufficiently intelligent to figure out a couple of these mistakes. I think by and large these are some of the glaring mistakes that I found, but if you find something else, just let me know, I will try to rectify them. And if you are terribly upset by any of these, I apologize. So, let us get on with our course. Let us get on with our module. Today we are discussing interaction of light with tissues. So, tissue is a heterogeneous matter, it is a heterogeneous mixture, and it is self-sustainable. So, unlike cell which requires a medium to carry it, something on which the cell will be put, tissue is self-sustaining, it can stand just by itself. So, it is like any other soft matter, any other organic matter, and therefore, the interaction of tissue as a matter with light in a way will be like interaction of light with any other matter per se. You will have refraction, you will have reflection, you will have absorption, and you will have scattering. Now, the difference with respect to, obviously you will have transmission and all of that. The difference with respect to the other materials, other matters, metals or semiconductors or glasses, with respect to soft matter or soft tissue in this particular example, is that the most prominent effect here is scattering. More than refraction, reflection or absorption, although you will see that these do play a significant role, these do play a significant part, the fundamental or the most important effect of light versus tissue interaction is the scattering. Why? Well, they have large number of scattering centers. Remember what was the definition of scattering? There will be a difference in refractive index at a length scale below the wavelength of light. So, a tissue is made up of several cells, several thousands of cells. Each cell have their own different membranes, organelles, there will be in extracellular matrices, all of that, all of those things that makes a tissue work or tissue function, and thereby they have several separate heterogeneous mixture of this scattering centers. They are not uniformly distributed, they are not homogeneously distributed. So, different parts of the tissue show different cellular arrangement, it can show. Each cell will have their own different cell organelles, centers which will be which has the capacity to scatter light, and therefore, the most pronounced light interaction that you see in tissue, the most pronounced optical mode that you will face while you are looking at a tissue either under a microscope or any other type of characterization method, light based characterization method, that is, you will encounter scattering mostly. All right. So, let us discuss this part. Absorption is important, that will come in a moment, you will see. Refraction and reflection we are going to skip for the time being, because these effects are not as pronounced as scattering or absorption. So, let us go with the scattering. So, by this time you know that the light scattering in general, not specifically for tissues, but light scattering in general can be specified into two separate categories, elastic scattering and inelastic scattering. What's the difference? Elastic scattering, the incident and scattered photons are of the same frequency. Whatever photon comes, it gets scattered away. It gets scattered away. The direction of the propagation of the photon may change, but the energy remains more or less same. Inelastic scattering is where a different energy of the photon is scattered. A different energy of the photon is scattered. The prominent example we discussed last time is the Raman scattering, where you develop a vibrational excitation molecule, where because of the interaction between the photon's electric field with that of the electron cloud of the molecule's electron cloud gets modified, gets deformed, have a conformational change that results in the molecule itself to rearrange, and it moves to a different vibrational quanta, different vibrational level. So, Raman scattering is a primary example of inelastic scattering. The other one is Brillouin scattering. The Brillouin scattering is that instead of a when a photon is hitting a particular material, not just tissue, any general material, there is a difference, and phonons, which are basically vibrations, are generated. So, there is a incident photon, there is a reflected photon. The difference in between, the difference, the first one has in the Raman spectra, that the difference in the energy gap between photons have changed the vibration of the molecule. In Brillouin scattering, this difference, this gap between incident photon and reflected photon or scattered photon has generated a vibration. The vibrations can also be quantized, and the quanta of vibration are phonons. So, these are inelastic scattering. The elastic scattering is, well, very simply Rayleigh scattering and Mie scattering. Rayleigh scattering is scattered by particle of size smaller than the wavelength. Remember, this is very prominent in tissues, scattering of particles, because there are several intracellular components, cellular components, etc., whose sizes are usually less than the light with which you are hitting. I am assuming you are trying to hit the tissue with visible light, 400 to 800 nanometer, 380 to 780 nanometers. So, here the scattering depends on the inverse of 4th power of lambda, that is the Rayleigh's law. You know, in high school we learnt about why the sky is blue, or why it becomes red during sunset. That is because of Rayleigh scattering. The blue light is scattered all over. The scattering is inversely proportional to the 4th power of lambda. So, that strongly contributes. Rayleigh scattering strongly contributes, and obviously the scattering is both backward and forward. Hence, the blue color all over is the sky. You know this, why sky is blue. The same thing is happening here. Rayleigh scattering is quite prominent in tissues. When you are hitting the sample with a bunch of photons, the photons get scattered according to their wavelength, and they scattered randomly all over the place, both back side and front side, forward and back scattering. Mie scattering is scattering by particles with size comparable to wavelength. So, it usually become more prominent elsewhere, or it does not depend on the intracellular components per se, and it has weaker wavelength dependence. You have weaker wavelength dependence, and it is mostly forward scattering. In tissue generally you see Rayleigh scattering. Raman scattering is, well, you obviously do Raman on tissues and thereby see change between tissue sample A and tissue sample B, or tissues which has undergone some kind of mutation, some kind of a change. So, that is seen. Brillouin scattering is coming up, but I am yet to actually get myself convinced that Brillouin scattering can be used for better imaging. Maybe I will be proved wrong in near future pretty soon, but overall it is Rayleigh scattering that affects tremendously tissues, along with a bit of all of these to a varying extent. Okay. This is the general type of scattering that happens, and when we particularly talk about tissues, it is this bit of all three. Okay. So, let us discuss a little bit about so what. So, when light enters or light excites or light hits upon a tissue, the tissue sample scatters the photon. So what? You can very well ask. Well, whenever the scattering takes place, whenever the so what part of so what, several chain reaction can follow. Remember, the major point that I raised is that in biological matter, especially photobiology, a chain sort of reaction, a domino effect. Remember, in a domino, when you hit one of those dominoes, all of them falls because of the chain reaction. So, whenever light enters biological matter, it has a capacity to give rise to series of subsequent steps, subsequent chain reaction. One might involve direct light hitting the substance, light hitting the material, cell or tissue, but it gives rise to sets of events, sequence of events one after another that result in something significant, something important that we need to see. So, what are the so what part of scattering of light with tissue, right? What happens actually when light has come, hit the tissue and got scattered away. So, two major processes, radiative and non-radiative process. Radiative process you already know, I discussed fluorescence. Tissue has materials, tissue has components that can autofluoresce, right. Fluorophore is usually a material, usually a tag, sometimes a label which is specifically made to absorb a specific light, which will then emit another light of a larger wavelength, lower frequency, that is specifically done to look at microscopy. But then there are certain material, endogenous material, native material, which are present inside a tissue that when hit by visible light, i.e. when you are looking at them using a visible stereo zoom microscope, they start fluoresce. Usually these intensity of autofluorescence is comparatively less, but remember, if you are actually working on fluorescence microscopy and your filter is not good, or you are trying to look at very feeble radiation, like you want to detect one single antibody, one single virus, a specific protein, or you are trying to look how the nucleus is behaving, then all of these combined effect of autofluorescence does interfere and may produce noise or may produce artifacts. Though most fluorescent microscopists that I have spoken with, they claim that well, that's what develops your skill set, right? What's the fun if everything is perfect? So let the tissue autofluoresce. I know several tricks and tips up my sleeves. I have several of these which will take care of autofluorescence. So that's going into a different topic. But remember, autofluorescence is an endogenous fluorescence. It is because of several particles such as several proteins, NADH, few nucleotides here and there. They autofluoresce. There is a beautiful picture in Wikipedia about autofluorescence of banana peel. You know, banana, you take it out and you hit that the outer layer of banana, the yellow layer of banana with different light, and you will see just looking at different visible spectrum of light, it does start emitting, or it does start showing fluorescence effect. We will be discussing FRET and fluorescence microscopy in the bioimaging part. So, I will keep the autofluorescence part for that. Though this is not very significant, you must be aware of what autofluorescence can do. Let us discuss the non-radiative process. These are the processes where, as the name suggests, photon is not generated. So, the light is getting scattered, some amount might get absorbed. Remember, scattering and absorption are analogous processes, and it results in subsequent chain reactions. Usually none of these chain reaction emit a photon. Okay? Radiative is where autofluorescence happens, non-radiative where all of these different processes, different photo-induced processes, different light-induced processes happen, and none of them actually results in generation of a photon. So, let us look at each of them one by one. This is the direct effect of light on tissues. This is the direct effect on light on tissues, and thereby you have to know. I am pretty sure that medical science students know all of that. Life science students also must be knowing it generally, but for rest of you, this part is quite important. So, autofluorescence, as I said, fluorescence from various components of the tissue. The photochemical process is occurring at a very low optical power density. So, very low power can result in some kind of a photochemical process. previous class in when we were discussing light interaction with cells. Remember, we discussed a lot of these photochemical processes where photodimerization, photofragmentation, several things happen. So it is similar here, where different molecules can combine, different molecules can fragment, they can form isomers, they can form several other compounds. The one important thing that you have to understand, or you have to keep in mind with photochemical processes, that it is low optical power density, right. So even when your incident light is of low power, you cannot guarantee that no photochemical changes are taking place. No photochemical changes are taking place. So you have to be aware of that. There is a possibility that specific wavelength, even at low power, low intensity, can result in a chemical change inside the tissue, and thereby whatever you are seeing is because of your observational effect, right. It is not something native to it. You have changed the tissue while putting it under the microscope. It is your work, it is your effect. You have done that. So that might cause problems when you try to make big discoveries that this thing happened or that thing happened. So be aware of photochemical processes because of the low optical power density. Let us discuss thermal effect. Let us discuss thermal effect. This is, I think, they teach you at first year in medical school, but intuitively rest of us already know. Thermal effect is when light gets converted into heat. Light gets converted into heat when light falls into a tissue. This conversion of heat happens either by intersystem conversion or interstate conversion. Remember this internal, sorry, beg your pardon, either internal conversion or intersystem conversion. The singlet state and triplet state, where in the fluorescence part where I discussed that in the same band, in the HOMO band, it can falls from lower level to upper level in the same band, and this is non-radiative. This is by vibration, this is by heat. And intersystem conversion, where it goes from a singlet state to a triplet state, there also it's non-radiative. No photon is generated for this transfer, it's just heat or vibration. So thermal effect is a direct conversion of light into heat. Thermal effect is a direct conversion of light into heat, and it depends when you are illuminating, usually when you are illuminating with a high radiance. When you are illuminating with high radiance, it is kind of intensity dependent, but it could also be wavelength dependent, depends, but most of the time it's not specific, and generally it is, I am not specifying it, generally it is wavelength independent. Okay. So suppose a very high intense laser, your body absorbs different frequencies, different photons, but say a very high energy radiation beam, you put your hand into it, the wavelength does not matter, it is the heat generation, it is the radiation intensity, those things matter. So when an intense beam of laser or intense beam of light falls onto the tissue, several effects, several consequences takes place, right. These are usually wavelength independent, usually, mark my word, though if your body starts absorbing, you will see different problem, but usually wavelength independent and non-specific. Most importantly, this happens because light energy gets converted into thermal energy. You do not require a particular molecular orbital to populate. You do not require the electron to jump from one area to another area, one energy state to another energy state. The population inversion, all of those things are not required here. It is simply, to put in layman word, burn. This is simply when light is burning your body. There is a huge amount of vibration is happening, and it is resulting in thermomechanical changes in that part of the tissue. There is no emission required or absorption required. It's simply light is getting converted into heat, and that heat is doing damage. The first damage is coagulation. This usually happens around 60 to 70 degrees Celsius. See, I was about to misspoke as percentage. So, coagulation happens when 60 to 70, around these temperatures, 60 to 70 degrees Celsius, where the cell membrane ruptures, organelles come out, and basically the tissue undergoes necrosis. Necrosis, fancy term for death. Cell, well, I won't say cell death, but the cell suffers necrosis. So, coagulation is at around temperature. So, if your light beam is ready to, if your light beam is able to increase the temperature of the tissue or the cell just by simply passing energy, just by simply vibrating, increasing the temperature to 60 to 70 degrees Celsius, the cell membrane can rupture, the organelles will come out, and the tissue undergoes necrosis. The second one is vaporization. Vaporization, where the temperature of the cell or the temperature of the tissue raises to 100 degrees Celsius because of light, because of intense laser radiation falling into it. Vaporization basically converts the water that is present in the cell or tissue to vaporize, to convert into steam. Pressure will build up because water is converting into steam, steam is gas, gas will have pressure, the pressure will build up and it will rupture the tissue. It will rupture the tissue and you will have problems, you will have breakdown and the tissue will undergo some kind of a damage. So, vaporization is at 100 degrees Celsius, water is converting into steam, easy. Carbonization. Carbonization usually happen at 150 degrees Celsius. This is where other molecular compounds simply break away and the tissue is charred, the tissue becomes black, it's like that very hard black color surface. It usually happens when we burn food. You remember when we actually burn food, or some of you who are non-vegetarian, sometimes you might see that if you put meat, chicken say for example, in oven or in tandoor, and if you have kept it for a long time, the entire food or the entire chicken become very hard, very crustaceous black color. That is carbonization. That is basically carbonization, usually 150 degrees Celsius or above. This is where rest of the molecules either detaches or fragments. It's just the carbon remaining and the carbon from strong chains, and it's that blackish, very hard crustaceous thing. That is what is happening at around 150 degrees Celsius and more. The melting process is quite important as well. We will be discussing melting at tissue engineering part. Melting is where the temperature, because of the presence of the light of the tissue, goes above the melting point of several different molecules. So, your protein will melt or your cellular structure will melt, and that obviously causes problem, but you will see in tissue engineering, you will see in subsequent chapters that we do utilize this melting of tissues to for disease removal, right? Think about a tumor remover. Can we utilize any of these thermal effects for removing a tumor? Okay. The other couple of effects that happens by light falling into tissue is photoablation. Here there is a direct breaking of cellular structure performed by high energy UV radiation. This shows similar effect as that of thermal ablation, the thermal effect. But remember, the main difference here is that in the previous case light got converted into heat and that heat caused problem. Here there is no conversion per se, there is no heat being generated, it is just that high energy radiation, UV radiation. The UV radiation itself is energizing the molecules, the electrons, everything inside the cell which forms part of the tissue, and thereby there is a breakdown, there is an energy, a huge amount of energy is being absorbed, everything goes into a breakdown condition. The cellular structure actually breaks down by high intensity energy radiation. So, this basically photoablation you will see when a human being is exposed to radiation, radiation poisoning in nuclear power plants, or God forbid in nuclear bombs, or those kinds of accidents where high energy radiation simply breaks down your tissues. Do not think about, or you do not have to wait to light getting converted into heat. The light itself with its energy is able to break down your tissue. The energy of the light, energy of the photon is enough to generate cellular structure damage. The plasma induced ablation usually happen when a very short pulse of light, femtosecond laser, femtosecond pulse or picosecond pulse with a huge power, say 10 to the power 11 watt per second or 10 to the power 7 watt per second, for a very, very small period of time, for 1 femtosecond, 1 attosecond, 1 nanosecond, hits a specific area of your tissue. Okay. You have a laser, a pulse laser source, a continuous laser source is like this, a pulse laser source will be like that. So, this pulse, the existence of this pulse is for few picosecond or few nanosecond, but in that nanosecond it has packed a huge amount of power, it has packed a tremendous amount of power. So, what happens when that pulse hits your body or hits a part of the tissue? Here plasma is generated, a direct breakdown of dielectric happens, there is a intense ionization, it is localized. So, light is forming here, there is a localization, and an intense plasma is formed, it got ionized. So, that much amount of power at that small area of time with that high that small period of time results in the molecules which forms the bond, which forms proteins, which forms nucleotides, which forms cells, they get ionized, i.e. their electron and the nucleus simply separate from one another and you have a plasma cloud. That plasma cloud, high energy plasma cloud can produce ablation. Okay. Photo disruption is a similar thing, but the difference of plasma induced ablation to photo disruption is a result of this formation of plasma. So, a plasma is forming locally, but it has generated something, a chain reaction that is happening somewhere else, meaning the plasma has energized either the liquid nearby, it could be water, or it could have energized carbon dioxide, any kind of gaseous matter that was present in cellular or subcellular structure, and that gas or that steam has traveled and produced some kind of a mechanical shock wave, right. So, it is like that bomb, it is like the bomb hitting at a place and the shock wave is moving at a different area. The shock wave is causing a problem. Here the center of attack, the center of hitting, that gets charred and ablated and everything. Ablation simply means destruction, but this intense hit produce a shock wave, a thermomechanical shock wave. That shock wave can travel to a different part and that can cause damage. It can generate steam, it can generate carbon dioxide, and it can cause significant amount of damage. While we were discussing the thermal part, while we were discussing the thermal effect, here it needs to be, I forgot to mention that it can be both local as well as non-local, meaning your the temperature is raised at a specific point, but say for example, the blood vessels underneath are carrying the heat to a different area, right. You have a heat falling onto a particular area of your tissue, the temperature of that is rising, it is causing problems at that area, but say you have underneath several blood vessels, the blood vessels can carry that heat to other areas, and depending on how much heat it has been able to carry, you can see problems or ablations happening at that area. So, it is not just the thermal process is local, it can go in different areas as well. So, that is more or less the end of the topic regarding interaction of light with tissues. We discussed about previous classes cells and molecules. Today I discussed light scattering in tissues. Remember, scattering is the most important part, and some of the light induced processes in tissues. this happens to my reference, and in the next class, in the last class, I will talk about a very interesting topic of mine, a very nice topic where I will give a specific example of light interacting with biological matter, okay, the essence of biophotonics. So, thank you very much.